We would like to show our respects and acknowledge the Bedegal people who are the traditional custodians of the land on which this meeting takes place, and to elders past and present.
Okay, let's start. Good afternoon, everyone. Thank you for coming here to our seminar about single-cell RNA-seq service at Ramaciotti Centre. As some of you might know, Ramaciotti was pioneer in offering a 10x genomics platform, but now we are excited to relaunch our single-cell RNA sequencing offerings under a new branding, mostly focusing on non-microfluidics technologies. Apart from Illumina single-cell RNA-seq, we also now support Parse, Evercode, and ScaleBio single-cell technologies. But today, our seminar is focused on Illumina single-cell prep that is based on PIPseq technology. And to begin, I'm pleased to introduce Ivonne Petermann. Ivonne is executive specialist in emerging technologies at Illumina. She will walk us through the science and innovations behind the Illumina single-cell RNA method. And after that, we will listen to talk from Andrew from Quinlan Lab, from Babs, that he is basically presenting us his work using PIPseq technology, which was actually done in our facility. And also, before we start, I'd like to thank you, Illumina, and their team here, Sarah, for sponsoring the event. We have afternoon tea, so please stay around after the talks to chat and network. So without further ado, I just leave it to Ivonne. Thank you.
Thanks, Firoze. Is it better if I stand over there? Doesn't matter? All right. Okay, good afternoon, everyone. My name is Ivonne Petermann, as Firoze introduced me, and I'm happy to be here today to give you a quick introduction on the Illumina single-cell sequencing technology or kit. Some of you might have heard about the technology. It is based on Fluent PIPseq. That was distributed by Chameleon Biosciences here in Australia prior to the acquisition by Illumina. So now you will see it as the Illumina single-cell 3' RNA prep. Unfortunately, a bit of a mouthful. Quick introduction. So with single cell, it's been around as a technology for a while. There is multiple different types of single cell technologies out there, especially some of the microfluidic space like 10x that have been establishing themselves really well in the market. But there is still, due to certain limitations, a barrier for more labs to get into single-cell RNA-seq due to cost, due to the acquisition or the necessity of microfluidics instrumentation, relatively rigid sort of kit packages and sizings, as well as sort of a fixed cell number that can be used. Etc. So all those limitations can hinder, a single-cell RNA-seq experiment, and this is where Illumina comes in with the new 3' RNA kit, addressing some of these issues. So there is no microfluidics instrumentation required, just a small little starter kit, as we call it, comprising of a vortexer and a small PCR machine. It's called a dry bath, and that's it. And you basically perform your reactions on these two instruments in tubes. So really easily scalable if you are working with very few cells. Some other technologies are really restrictive in that regard because there is a minimum relatively high cell number often required as a starting point. With PIPseq or Illumina single-cell RNA-seq, you can start with as little as 100 cells per reaction. And it is extremely affordable, even at list price. It's much, much cheaper than, compared to 10x, for example, out there, with the added benefit of having a lot of flexibility. So as a quick overview, I know if you can bring this into your own lab by using or acquiring that little starter kit yourself, or you can use the local service providers here that have this workflow set up. And essentially, you are capturing or encapsulating single cells or nuclei in suspension. So it is an emulsion-based technology, however, without a microfluidics instrument. So you are pipetting your cell suspension or nuclear suspension into a tube that contains our particle templated beads. And then you vortex that tube for 2 minutes, and that's it. What you've achieved now is created an emulsion capturing all your cells or nuclei efficiently. This can be scaled up simply by using larger vessels, so larger tubes, all the way going up to a 50 ml a tube for the large 1 million cell per sample kit. So you can pick and choose depending on what you want to do. Each of these reactions per kit can also be done and taken out individually. So no complex thinking and calculations of making sure you are utilizing the chip and the reactions optimally to essentially get it most cost effective, but also use up all the reagents. So this is really scalable and basically pick what you need kind of packaging. The workflow, for those of you who are already doing single-cell RNA-seq in one way or another, looks quite similar. So it begins with capturing the the cells or the nuclei in the emulsions, as discussed in this short vortexing step, and then you're creating PIPs, so particle templated instant partitions. Following that is a lysis step, so we are lysing the cells and capturing the mRNA on the oligo-DT beads, coated beads. So it's a 3' total RNA capture method using these oligo-DTs, so there is no bias, there is no large probe-based kits involved. You're capturing unbiased polyadenylated RNA molecules. That step only takes about 10 minutes of hands-on and setup time, and that is minus the time you need to prepare your cell suspensions and nuclear suspensions. And then you've already reached the first safe stopping point. So at that point, the mRNA is in the particles, in the PIPs, stable and safe for up to 96 hours at 20 degrees. So that allows you to batch additional samples. It allows you to transfer it to a service facility or to a collaborator, or just simply store it to run it at another time following the weekend, for example. So once you continue, also very quite straightforward, you perform cDNA synthesis in the beads, and then continue on with library preparation to make Illumina compatible sequencing ready libraries. For the analysis, so it will fit on multiple different sequencers. I've got a couple of slides on that in the end as well. But for the analysis, Illumina also brings the entire end-to-end workflow. So we have the Dragon single-cell RNA pipeline that comes free with your PIPseq or Illumina single-cell RNA-seq package. As I said, it does the secondary analysis, and you can then plug the results from that into PIPseq Flow. Again, we've acquired PIPseq last year or 2 years ago now almost, and have adapted this pipeline to analyze the Illumina single-cell RNA-seq data. It also does other types of single-cell RNA-seq data sets, so it is not limited to this, but that is an option currently available, and in the future we will have Illumina connected multiomics as an analysis option coming up towards the end of the year. This will then also handle multiomics data like spatial data, for example, so you can combine different analytes from the same sample in one analysis platform to visualize and play with the data and get your graphs and differential gene expression analysis, etc. So this slide highlights the sort of sequencing throughput, sequencing capacity you need to consider for this. I guess the most important fact is you need to sequence about 20,000 read pairs per cell or nucleus captured, and then perform, depending on how many samples you have, how many cells you've captured, the appropriate sequencing run. And then there is a few additional recommendations listed below regarding read lengths and the library structure to make sure you're setting this all up correctly to sequence. But sort of at the basic level, the Illumina single-cell RNA-seq libraries can be analyzed with a 100-cycle kit or sequenced with a 100-cycle kit. I know we are hearing firsthand some data today from Andrew around the performance of this kit. I wanted to quickly point out a couple of things in the next few slides around this. To start with the kit format that I mentioned to you earlier. We have 4 different formats available at the moment, and they differ in the number of cells that they're optimized to capture per sample. So you can pick and choose depending on how many cells you want to capture, which kit might be best suited to your application. You can also choose one of these larger kits to multiplex samples together using #oligos that is compatible with our technology as well. They do vary slightly in the numbers of reactions that are supplied per kit, so just be aware of that. So the smallest kit, the T2, lets you capture 2,000 cells and you get 8 reactions for 8 samples if you don't multiplex. And the large kit, the T100, lets you capture 100,000 cells per sample and you get 2 reactions. The next focus on this slide I want to bring to efficiency and capture. We often get asked how efficient actually is it. So we have seen up to 85% cell capture efficiency. Typically we see this in PBMCs. The efficiency is slightly lower if you go with complex samples, or you use cell suspensions derived from tissues, etc., or if the cell viability is a bit lower than we would like or you would like. But sometimes you don't have a choice. You have to run a sample even if it's if the cells are suboptimal. So that will have an impact on the capture rate. We have a very low multiplet rate, so the rate or the amount of PIPs that you generate that have more than one cell or one nuclei in it, these typically are lost because you can't differentiate the transcript that are captured from being derived from 2 different cells. So those typically tend to be data that are getting filtered out. The workflow itself, time to library, is a good 2 days workflow in the lab, but Andrew will probably also mention this, how long this takes. And for PBMCs, on average, we see about 1,800 genes per cell, or about 4,000 or more transcripts per cell. So just as a rough indication, again, this will obviously vary depending on your input cell line or cell or nuclei. Briefly, some data around the low multiplet rates that we have demonstrated, comparing this to the 10x GEMX kit. And when we look at comparing the Illumina single-cell RNA-seq data with the GEMX kit as well, we see, or we look at transcripts per cell captured versus, well, not versus, as well as genes per cell captured. And you do see there is they look quite compatible. There are differences. However, the consistency between experiments is quite high, and also the consistency between the cells that are actually culled between both kits is really high. So both kits cull all the same cell types within the sample. We have seen in our comparison that was conducted with, in collaboration with Harvard, when you sequence even less cells compared to the 10x, that we are identifying more cell types, so 56 cell types in this case compared to 52. So it can actually overachieve compared to GEMX. We've also seen extremely good performance in nuclei, specifically for mouse brain. This is a graph or two graphs where we are actually looking at mouse brain cells that were run, 155,000 were captured here per reaction. And we see a large number of cell types that were discovered or analyzed, detected in the mouse brain. So for people that are working with this type of sample, they're really excited about this, because often with other technologies, they miss certain cell types, and microfluidics also tends to be a problem for large cells or oddly shaped cells. So you sort of don't run into the same problem with PIPseq or Illumina single-cell RNA-seq. And then again, on the right, you can see sort of the median transcripts that are detected per cell is actually also extremely high compared to, this is actually not GEMX data, this is compared to 10x version 3.1. In addition to 3' standard whole transcriptome analysis, Illumina single cell allows the analysis of additional modalities. We can detect extracellular membranes or cell surface membrane proteins utilizing the BioLegend Total Seq-A products that is compatible with the utilization or in combination with our supplementary enrichment amplification kit to pick up that information from the oligonucleotides on the antibodies. You can hashtag, as I mentioned. This is also done through the SEA kit in addition. And you can do certain additional things like custom amplicons, etc. And what's also available or possible with the implementation of the SEA kit is CropSeq. So you can already do a CRISPR pooled perturbation experiment, measure the transcriptome and pick up the guide RNA that's expressed per cell. However, we will actually bring out a kit shortly that allows us or you to do this at the same time. So you can measure the guide RNA directly in the same cell, in the same reaction, with slightly changed beads that we are using in this kit. This is an example of what's currently already possible, a publication on this, and this is a poster that for those of you who are interested, I can send to you, where we are demonstrating a prototype, a data set generated with our new CRISPR kit that's coming out on the million cell scale as well. So really exciting data here. And this is an early access now, so hopefully we'll be able to bring this out to you soon if you're interested in CRISPR. And in the last few minutes, I wanted to go over some of the data analysis features that we have built into the end-to-end solution. I know a lot of you actually always tell me, I like my own pipeline. Can I do this? Yes, you can. So you can take the data that's analyzed through, for example, our Dragon single-cell RNA pipeline that comes directly from the sequencing run. You can take this in your own pipeline, or you can use PIPseq flow. So there's options there for you, so you can choose which one you prefer, or also use, I guess, both side by side to start with as a comparison. So to bring this together, Illumina wanted to make sure that it's all sort of in one go. You have everything from the RNA library prep to the data analysis. And we've listed here some of the features that the Dragon as well as PIPseq flow informatics and software tools perform. So the secondary analysis that Dragon performs is some of them are doing mapping and alignment, doing cell and gene counting, looking at our newer version or newer way of using transcript counting. So we don't have UMIs anymore in our workflow. We are using IMIs, so intrinsic molecular identifiers, that are being introduced after the cDNA amplification. And we are introducing sort of random cuts, random sites, and this is used in conjunction with our binning index, or BIs, to identify which of these transcripts were actually derived from a PCR amplification and which of the transcripts was actually the original captured in the cell. So it's a really good way of making sure PCR or amplification artifacts are being pulled out. It also generates basic UMAP images, it does feature counting, etc. And then you can plug this into the tertiary analysis platform, which is PIPseq flow, for visualization and for cell type identification, differential gene expression, pathway analysis, etc. So in the interest of time, I'm just going to go through some of the snapshots of the interface. It is relatively straightforward to use, so you don't need any coding experience to do this. The PIPseq flow works with, this is actually first the Dragon, a single-cell RNA pipeline giving you a cell by gene matrix, a cell sample identifier, a list, and a single-cell RNA metrics file that you can plug in. But then PIPseq flow is like drop-down menu type analysis, where you can pick and choose what you want the software to do with your data, and to also customize the views. So some of these things that can do are listed here. Again, I think for a non-bioinformatician, this really does a very good first cut analysis for you, and allows you to look at your data sets. And if you wanted to go dive deeper or utilize, pre-existing pipelines, you can obviously do that as well. So we have multiple different plots and charts that are being produced in this workflow. For anyone who is particularly interested in one of these features, or just to get a demo of the PIPseq flow, we can organize that via Teams, and we can also provide a free trial of the software if you want to give this a go with sample data. So just quickly, some of these snapshots of plots and that you can see, and how you can pull them out, configure them, customize them, so change colors, change views, overlay specific reference transcriptomes or genomes as well. And you can see here at the top sort of one of the first windows where you have a decision tree, what data are you actually looking at. So as you can see, it also handles bulk and microarray data, for example. You get quality plots, quality assessment, QC plots that you can look at, and you can obviously see the UMAP or TSNE plot, so it generates both, depending on which one you prefer or which one you like. And for cell type classification or cell classification using specific biomarkers, this is done obviously with the file you're supplying, but you can also change the way a cell is called by changing manually what you want this cell to be called by looking at gene expression plus biomarkers. So some of these features I think you'll appreciate in a live demo or when you actually play with the data and analyze some of this yourself, but it does give you a lot of options already in the way you visualize them and in the way you analyze the different present cells and the gene expression. And beyond cell type identification, you can also look at pathway analysis, for example, where some of these genes that are differentially expressed, which pathway they belong to, for example. Yeah, so that is the part of the biological interpretation, which is all partly preloaded into the PIPseq flow workflow. So in the interest of time, I'm just going to finish going through some of these screenshots. Again, feel free to reach out. We can provide a demo to you from our bioinformatics team. And I think Andrew will actually show us some of the data his team has produced and how they've analyzed it. And we have multiple resources available for learning how to use these tools for single cell analysis as well. So with that, thank you for your attention. And I wanted to say Zara is here and we will be here afterwards for a discussion. If you have a project idea, let us know. We do have still a special available to purchase some of these kits through the center here if you want to give this a go. Thank you.
Yes. Okay, thank you, Ivonne. That was a great talk. Is there any questions? Yes, please.
Thanks. Yeah, thanks a lot. I was sort of curious about the PIPseq angle and what you were presenting there. So did I understand correctly that you still need the PIPseq construct at this stage and you need a polyethylated system? It's not yet possible to capture directly the guide itself, right? Okay, right. Thank you. And the data you said is going to be made publicly available of your pilot. Is that true?
But we should have the data available as well in the coming months prior to launch.
Yes.
Hi, is there any in your workflow of possible new adaptations for PIPseq? Is in the horizon any plans to adapt this for multi-omes, so doing ATAC-seq and single-cell RNA-seq simultaneously using this technology?
Yeah, that's a good question. So we have on the horizon to expand the offerings for our solutions. So PIPseq or CRISPR is going to be the first one. We have to decide between 5', which is also something that we get asked a lot, and that a lot of people want to use attack, and a combination of both. So we have not finalized what's going to be prioritized, but it is definitely a discussion.
Thank you.
I can't tell you what the timeline that might be.
more questions.
Thank you, Yvonne. You talked about doublets. PIPseq's unique in that it could potentially form halflets, meaning two pips for one cell. Has anybody looked at this?
That is a good question. I can check. I'm not aware of the data or that we have looked at it, but I can check.
It's the kind of thing that most people would just ignore, right? But then you can imagine that should be possible. And how could you detect it? Food for thought.